So after all the exciting talks and all the discussion, now we come to the last presentation, also one of the most interested area about therapeutics and treatment of Pompe disease, which is also talking about yesterday, tomorrow, and actually it's yesterday, today, and tomorrow, like the way forward. So, and the talk will be by Dr. Sheng. So, Dr. Sheng is actually the consultant neurologist at the Department of Medicine and Geriatric in the Princess Margaret Hospital in Hong Kong. Dr. Sheng was graduated in 1995 from the Faculty of Medicine at the Chinese University and of Hong Kong, and he started his professional career first in geriatrics and then later switched to neurology, and afterwards he completed his fellowship training and become a specialist in neurology. Dr. Sheng actually has special interest in inborn error of metabolism in adult patients. With the support from his hospital, he established the first designated IEM clinic in adult medicine in 2012, and that clinic was subsequently endorsed as the only designated adult treatment center for enzyme replacement therapy in lysosomal storage disease in 2014. So he and his team actually received referral from all the other units and all the other hospitals of patients with lysosomal storage disease in Hong Kong. In 2015, Dr. Sheng was also awarded a Hospital Authority Corporate Scholarship so he can pursue his advanced training in genetic medicine in Westmead Hospital, Australia. He's now a member of the Hospital Authority Expert Panel on Enzyme Replacement Therapy for Lysosomal Storage Disease, and he has also served as a medical advisor to rare metabolic disease patient support groups. Dr. Sheng also is a key member of the Clinical Research Center in Princess Margaret Hospital, the chairman of the center to oversee the training and research activity in his hospital. He also has been involved in several international multi-center clinical trials and contribute more than 50 publications in both local and international peer-reviewed journals. So without undue delays, it's your time, Dr. Sheng.
Thank you, Dr. Chen. This is my disclosure and also disclosure from Sanofi. I'd like to talk about Pompe because it is not just from a scientific view, and actually Pompe connects people together from patients, from families, from industry, from physicians, and from scientists. So if you read about Pompe, you notice there are many information may not be directly from the scientific view, but really true, fairly tell stories. So if you look at Pompe, you can look at it from different perspectives. It can be a devastating medical illness. It can be also something not related to us physicians, but related to the business field with this guy, Mr. Cowie, who is a father of the Pompe patients who try to find the treatment for the children. Finally, he funded the first enzyme research and got himself into the CEO in Genzyme, and later still now the CEO president in Amicus, and he finally this year got his first enzyme under his name from the Amicus. So it is really a showcase, very fascinating. So that's why I really found Pompe's disease is very special. And also the Dr. J.C. Pompe, the pathologist who first described the disease, his legacy is not just ended in Pompe disease because he is a national hero. He when Netherlands was invaded by German in World War II, he used his hospital laboratory to shelter the Dutch teletransmitters, but was finally found out and being arrested, and unfortunately he was later executed just before the end of the war. So his legacy is beyond Pompe. After discovering the disease, there's another person. You need to remember, Netherlands is very important in Pompe disease. And this group is just from the neighboring countries, from Belgium. The professor Duva and his team, and they are biochemists. They interest in carbohydrates. But incidentally, they found there are some intracellular particles with digestive properties. And when they investigating the effect of insulin in the liver cell, and they later call and name these particles lysosomes. This actually is the beginning of understanding of intracellular organelles, the lysosomes. And his colleague, Professor Hurst, who did not join the lysosomal journey, he continued his legacy in carbohydrate metabolism. He tried to find the enzyme for GSD3, but unfortunately, he failed to identify this because GSD3 also has muscle weakness, but he failed to demonstrate this enzyme is for GSD3, but the enzyme is deficient in his other Pompe disease samples. So with this knowledge, he later worked out that the enzyme only works in the acidic environment, and within the cell, lysosome is the organelle with the acidic environment. So he went on to prove this is an lysosomal enzyme. So actually, this is the discovery of the acid alpha glucosidase in 1963. And Dr. Hurst later published a paper defining the disease inside the lysosome with the example of Pompe disease. And this is the first illustration of lysosomal storage disease, which we now have more than 70 different lysosomal storage disease being identified with a collective birth instance of 1 in 5,000. So Pompe disease really has a very important scientific legacy in the disease and in the scientific field, and also the discovery of LSD. And you can realize LSD is still a very young disease because it is only described in the 1960s and 70s, and then rapidly, just 30 years after the first description of the lysosomal storage disease, we have the first enzyme therapy successfully started in Gaucher's disease. So you can see the development of the scientific field is very fast. And now we have many different enzyme replacement therapies and also new therapies available for different lysosomal storage disease. Another important figure from the Netherlands group, Dr. Van der Ploeg and her team was the first one who make attempts to use enzyme therapy in Pompe disease. They make use of the discovery that lysosomes need so for enzymes traveling into lysosomes, they need to have mannose 6 phosphate receptor engagement. Therefore, they modify the enzyme with phosphorylation and produce the first enzyme from the bovine testis into mice, into the normal mice, and demonstrate successful uptake of this exogenous enzyme into the muscle and the heart cells. The result was published in 1991. This is the first animal, the proof of concept in the animal study. So this is really the beginning of the story of the Coe's family, because after that, Mr. Corey pushed forward the human the trials in enzyme therapy. At that time, there were two groups. One is the US group, which had another important person, Dr. Chen Yunzhong, who is a Taiwanese scientist. At that time, he led the group in the Duke University. And another group, of course, from Netherlands. And there are two methods to produce the recombinant acid alpha glucosidase. The Netherlands group used the transgenic rabbit milk, and the Duke University group used the Chinese hamster ovary cell line. Both has been trialed in humans with success, but eventually it's the Duke group with the CHO cell line finally come into the mass production of the commercial enzymes, because it is technically easier to harvest the enzyme from these CHO cell lines. And because of the importance of Professor Sheng in Pompe disease, when he returned to Taiwan, he led the development of Pompe disease in Taiwan. Therefore, Taiwan is the first region to have newborn screening for Pompe disease. So this is all very related. And the first landmark clinical trial was conducted in the Duke University, led by Professor Kashnani. She's still very active in the Pompe field. They study the enzyme effect in infantile onset Pompe disease. And with a comparative study of 18 ROPD patients treated with glucosidase alpha at 2 dosage, and these patients all survive at 18 months of age. Their results are very, very convincing. You can see they reduce the death by 99%. So no death, risk of death or invasive ventilation by 92%, and the LV mass dropped by half beautifully. So this is just after 1 year, and because of that, the enzyme was rapidly becoming available for ROPD. And later, the result was replicated in the in many of the publications, with improved survival and ventilation-free survival. And we also know that if you treat the ROPD patients earlier, for example, being picked up by the newborn screening, then by clinical presentation, then the outcome is much, much better. This is from the Taiwan experience in newborn screening. But we also know the effect of anti-drug antibody, because some patients, if they do not have residual enzyme, which we call it cream negative, and then they will induce anti-drug antibodies, which will largely limit the efficacy of the enzyme. Therefore, the current practice is to test the cream status and to induce immune tolerance therapy before the enzyme therapy in those with cream negative. And we now also know that dose is also important for the benefit, for the clinical benefit. And now people are treating patients with higher dosage, both from the beginning and also when there is any clinical deterioration in ROPD. So these are all the experience in the past 20 years in the use of 15 years, should say, in the use of enzyme therapy in ROPD. And we also realize there is new phenotype developed in ROPD patients because they live longer. Those change, the cerebral change, which only developed at the childhood, was never seen when there was before the availability of the ERT. Now we are prolonging the survival, and now we face new problems which cannot become reversed by the enzyme therapy, because we know the enzyme cannot cross the blood-brain barrier. So the experience in ROPD, we have good survival, good ventilation-free survival, and also improved mobility. We have many experience seeing ROPD patients still mobile, exercising into their primary school age. We know the importance of age to onset of to start the ERT. We know the impact of immunogenicity, and we understand the dosage, importance of dosage in the ERT, which all have effect in the outcome. Few years later, the Netherlands group, they produced the landmark study, the LOG study for late onset Pompe disease patients. This group of patients, in the Caucasians, they are in the middle age, 40 to 50s. The improvement is not, of course, as dramatic as in ROPD, but they can still show improvement in the mobility and also stabilization of the respiratory function. And the longer term study also show sustained effect with sustained improvement compared to baseline, and also majority of the patients will perform better than the projected natural course. So, if you go for the longer term, you can see the effect of enzyme therapy probably will be maximally achieved in the first 2 years, and then gradually you still see a slow decline both in the ventilation function and also in the mobility. Does this improve survival? It is difficult to confirm, but from some of the real-world data using different statistical models, they tend to show there is a better survival in LOPD, but that was, of course, not as prominent and convincing as in ROPD patients. And just look back at those patients who has been recruited in the initial clinical trial after 10 years. We still see a lot of patients invariably will develop severe disease endpoints. That means from new ventilation to the use of non-invasive ventilation, and from mobility to become wheelchair bound. But there's still a large variation in individual response in the long-term follow-up. So our experience in ERT in LOPD, we know definitely it improves and stabilize of the muscle strength and respiratory function. When their quality of life improve, and possibly they could enjoy a better survival. But there is certainly unmet needs in the treatment of Pompe disease, despite our good experience in RPD and our similarly positive response in LPD, because invariably patients gradually deteriorate their strength and respiratory function over the years. Why do they, couldn't the disease be cured by enzyme replacement? One of the, of course, we talk about autophagic myopathy. We know the enzyme cannot reverse the autophagic buildup and clear the cytoplasmic glycogen accumulation. And this kind of autophagic debris will interfere with the enzyme trafficking into the lysosomes. So that, and also we know in the skeletal muscle in particular, there's lack of the abundance of the M6P receptors. Therefore, maybe this is the reason that the enzyme does not work particularly well in the muscle compared with the heart. So now this year, and actually last year, we have the second generation ERT. We have two products now coming to the market, vagosidase alpha and cipagosidase alpha. Both of them make use of the increase in the mannose 6-phosphate residues with the molecular modification of the original enzyme. And I just want to say, don't want to present the trial results, but want to say that both products demonstrate better patient outcome in the phase 3 trials over a wide range of different parameters and also the biomarkers. But we believe enzyme therapy is not the ultimate answer in Pompe disease, and the gene therapy should be a better alternative in the future. And it is also approaching and today. And gene therapy is frequently quoted as the ultimate cure in almost all of the hereditary disease, because it is a one-off treatment and there was no immunogenicity, and it is also CNS penetrable. And we now have different modes of gene delivery. It can be muscle targeted, liver targeted, because liver is an important protein producing cells. And also you can have in vivo and ex vivo. For ex vivo, it is to mobilize your CD34 cells and as being extracted and mobilized, and then was incorporated with the gene and then transfused back to engulf into the marrow, and then you can carry the enzyme to the different tissues. While in vivo, you have the direct injection or infusion of the viral vector together with the gene. I just want to alert you that there are already many genetic trials now active, and 2 of those trials are from China. Actually, nowadays, almost 30% of the genetic clinical trials are being conducted in China. So China actually is moving fast in genetic therapy. I want to share just one case. This is the first case of the gene therapy in Invita Pompe, because the company was just licensed to start the gene therapy for RPD last year and LPD earlier this year. And this, I believe, is their first case which they demonstrate the 3-month outcome. This is the first case of RPD. Patients probably 2 to 3 months of age. The patient had received 2 doses of enzyme before being put on the gene therapy. And after gene therapy, you can see the GA level without enzyme has been maintained. And most importantly, you can just visualize the regression of the hypertrophic cardiomyopathy in this young baby over the 3 months period. And actually, ROPD is actively recruiting patients in China. I don't know whether our next patient can be benefited into the clinical trial. The trial was centralized. The trial center was in Beijing. And we also found there is a side product of the genetic therapy is to improve the immune tolerance when given the enzyme therapy. This is shown in the Deuce University group. And so I want to end with this slide with a quote by Mr. Corey, talking. His life is about fighting. So he said that as human beings, we are defined by our core, at our core, by how we respond to hardship. Good doesn't just happen. So you want to have good things to happen for yourself, you have to fight. So from being an incurable, devastating, and fatal disease to our then focusing on better survival, better strength, and better quality of life, we already see lots of love, hope, and excitements. But truly, we still have unmet needs. But with the courage seen in the Pompe community, physicians, scientists, patients, and also the community, together we fight and we believe we will make miracle again and again. Thank you.
Thank you so much, Dr. Sheng, for a very exciting and inspiring talk on the therapeutic aspect, covering all angles. Is there any question from the floor? Okay. Can I just ask the last question? That I see no question from online. So you mentioned it's exciting to hear there is a new generation of enzyme replacement therapy, and there's improved markers that is being demonstrated from the clinical trial. Can you explain a bit of this? What are the markers they are looking into?
Right now, they believe the HAS4 during HAS4 will be a good marker, because in the at least in the LPD, of course in LPD you have obviously a good clinical improvement, but for LPD, for individual patient response, there are a lot of variations, because we believe a 6 milliwatt test is not very good, and FVC probably is not sometimes not very reproducible. But the biomarkers always being relatively static over time. And therefore, last week when and Nancy come to Hong Kong, she also advocate on the use of biomarker. So we believe henceforth now has been a more reliable biomarker. And of course, sometimes we can use CK, but CK probably is not very specific, but henceforth probably is a good, better biomarker.
Okay. So is Nancy Chai, professor from Taiwan? Is it? Is
Yes,
Nancy
yes,
here?
yes,
Oh, okay.
yes.
Thank you. So I think because of the time, I cannot allow further question. Can I now actually invite Dr. Sheng to join me to be the co-chair to have the closing remark of today's meeting?
Thank you, Professor, Dr. Chen. I would like to thank Professor Nicino and also our local speakers, Victor, Ronnie, and also Jean, giving excellent talk and very comprehensive review on the Pompe disease from diagnosis to the imaging to the genetics to the histology. I think really Pompe disease is a very important, especially with a lot of advancements in this area, and we will again work together because this is not we cannot work on our own. We need a combination of different expertise to work together to make this disease management and diagnosis improve, further improve. And we should not stop in the Pompe disease because we understand a lot of hereditary muscular disease do require similar collaborations. And we hope that with these kind of meetings under the leadership of Sophie, the Neuromuscular Society, wonderful society with a lot of expertise focused on muscle disease, we will culture our next generation colleagues and to focus on the neuromuscular disease, both from clinical aspect, from laboratory aspect as well. Thank you all for attending this symposium, and I wish you a happy and enjoy your weekend. Thank you.